Okay, so good afternoon, everyone. We'll start with week 9 of the course Host Pathogen Interaction by Professor Himanshu Kumar from IISER Bhopal. So, I am Rishabh, and in this lecture video, we'll be discussing about how a virus interact with the humans and how the immune system actually helps to overcome that and how a virus actually evades the immune system. Okay, so starting with the viral pathogenesis. Okay, so basically there are two three things that we need to study when we are seeing the host pathogen interaction of a virus with the host. Okay, so first is how a virus actually evades the immune system. Then the second component is the replication cycle for the virus. The third component comes from the host, that how the immune system of the host tackles this particular load of the virus. Okay, so for that, we'll start with the basic things that how a viral pathogenesis takes place. Okay, so it basically results in change in the physiology, resulting in deviation of vitals and culminate to the clinical symptoms due to viral replication. So, whenever a virus actually infects a host cell, then the virus basically takes over the machinery of the cell. So this machinery give rise to the production of newer virus particles. So based on this, what will happen now? Based on the replication and how much the new virus particles are formed, there will be symptoms. Okay, so these symptoms basically tell us how the vitals and the clinical symptoms will appear after the infection of this particular virus to the human body. Okay, so this is what is termed as viral pathogenesis and what we'll study today. So the infection can be productive, abortive, or latent or reactivation. Okay. So if there is acute infection, that means that is actually a very short-lived infection. So acute means that the intensity is high, but the time frame is very less. Okay, that's why you can see here the in for the infection and the on the x-axis we have the time frame. So whenever there is an acute viral infection, then the peak for the infection goes up to this point and then it comes down. That means the infection now fades away. Okay. For the latent infection, what happens? There is one major infection. Now, there can be a chance of reactivation of these infections. That means the virus is staying in a latent phase inside the body. So, whenever the virus gets the favorable conditions, it can reactivate itself and cause the symptoms, as you can see in these two points. Then, if there is progressive infection, then for the first time, the intensity of the infection actually goes up. And then, followed by this, what happens? There is increase in the intensity of the infection with time. So, that is progressive infections. That means the virus that can be inferred from this graph is the replication of that virus is slow, so that whenever the number of viral particles will increase, this would increase in the symptoms and the viral infection. Okay, then we have T, that is chronic or persistent infection. Chronic means every time long term. Okay, so when the intensity of the infection increases, then that is not completely removed from the body, but it leads to the continuous showing of those symptoms because the viral particles are present inside the body. So that is termed as persistent infection. Now, if there is failure to clear all the evidence of the infection, so that means what happens there, for the first intensity of infection it's there, but then the some amount of viral particles remain inside the body. Okay, so that's why the the full symptoms do not disappear from the body because some of the viral particles are left inside the body. So that is why there is a failure to clear all the evidence of infection. Okay, so these are the various pathogenesis pattern that follows in a particular host that is infected by the virus. Okay, so moving on. So these are basically the simple terms how a virus basically infects a how a virus infects the host and how host can actually overcome that. So this is the virus that infects the host, and after this what happens, there are certain cytokine factors, epigenetic factors, or the viral factors that takes place, and those host genes can either resist or increase the viral infection. Okay, so because of those, the body always tend to resist the expression of those genes that are there from the virus because those are foreign genes. For that, they'll actually initiate a cascade of pathway that will lead to multiple outcomes. For example, I am giving you one example of IFN gamma. Okay, so this IFN gamma basically leads to increase in the inflammatory response. Likewise, there will be multiple responses against those viruses because of our immune system. So that is what it is shown here. Because of the different pathways that are initiated because of the viral infection, there would be different outcomes from those pathways. Okay, those sorry, those outcomes can be leading to similar conditions like the inflammatory response or the activation of the T cell or the macrophages. So that can be a different case, but the outcomes can be different from those pathways. Okay, then the pathogens or viruses basically, why do they infect the host? Because they can survive and multiply in the appropriate host. How is it possible? They find a nutritionally compatible niche in the host so that they can get constant nutrition. Then they can replicate using host resources. As you would know, based on the initial classification, virus is basically non-living outside the body, and it is living inside the body. They colonize in the host. They avoid or subvert or circumvent the host innate and adaptive immunity so that they can overcome the particular immunity and they can colonize or they can take over the cellular machinery. Okay, then the strategies that they use is not to kill the host. If the host get kills, then it can be highly infectious, known as facile transmission. Okay, that means whenever they are not able, they do not kill the host. If they kill the host, that means that can be deadly virus. Okay, and those viruses can also infect the healthy individual. Okay, so this was the basic introduction how a particular virus pathogenesis takes place. Now we'll be moving on to certain specific areas where the viruses actually the virus actually go through. Okay, so first we'll see how the body reacts to the virus infection. So we have the innate immune responses. So virus, because it's a pathogen, it will have the recognition, it will have PAMPs. Okay, pathogen associated molecular patterns. Now these PAMPs will be recognized by the recognition receptors in our body. Okay, so there is direct detection of the viruses by the PRRs. So the viral signatures are viral nucleic acids, because the viruses actually include their nucleic acid into the host cell machinery. So they'll need it for replicating inside the host cell. Their long double-stranded RNAs containing 5' triphosphate, and unmethylated CpG motifs in the viral DNA gene. Okay, so these are the common recognition receptors from the viruses that the body recognizes. Additional factors help determine the viral origin. So, because there would be different origin of viruses, because there are different classes of viruses that infect our body, so there would be some additional factors related to the PRRs that will be identified by our body depending upon the type of virus that infects the body. Okay. So what happens is, sometimes, as we can see from the literature, there is the host nucleic acids are not found in the endolysosomes, but the host DNA. So as we can see in the cytosol, okay, directly the DNA is not stable. This is case of the host cell. Okay, so what happens is, whenever they're in the cytosol, this is the machinery that has been evolved in case of the host cell. So if there is any naked DNA that is present inside the cytosol, that means it is not its place. So the body tend to degrade it from the host cell machinery. Now the virus need to deliver this DNA to the nucleus itself. That's why in different literature it has been seen that in the endolysosome or the cytosol, only the viral particle are formed. Viral particle are seen, but not the nucleic acid. Okay. Now this was the viral signatures that help in direct detection of the viruses by the PRRs. Now, there are indirect detection methods also, because there is alteration in the normal cellular processes. For example, the acute decline in the host protein synthesis, altered activity of ion channels and ER stress. These are not fully categorized although, but wherever there is dysregulation in the protein synthesis, ion channel activity, or increase in the ER stress. So that means that can be a sign that a viral or a pathogen has taken control over the host cell machinery. Okay, so these are the common symptoms at the molecular level that shows us whether the virus particle has actually taken over or not. Okay, so based on this recognition, the innate antiviral mechanisms activate. That means they try to secrete the pro-inflammatory cytokines from the NK cells or the macrophages. Okay, so the cytokines that are pro-inflammatory are type 1 and type 3 interferons, IF and gammas basically. Now, after the innate immune system actually recognizes these, the adaptive immune system comes into play. Okay, now this adaptive system is basically antigen specific and it provides a long lasting viral immunity. And here, majorly cytotoxic T cells and the NK cells are the major players that actually help in sustaining the immune response against the viruses. Okay, and there are neutralizing antibodies also. So these neutralizing antibodies basically help in stopping the infection, and all these mechanisms including CD8 lymphocytes, the NK cells action or the neutralizing antibodies, all these mechanisms ensure inhibition of the viral replication, viral gene expression or virion assembly. Okay, so this is how our immune system actually reacts to the viral infection. Okay, so any doubts in this particular slide or till here? Okay, so let's move on. So now we'll see how an innate immune system actually reacts to the particular virus. Okay, so as we have studied in the previous lectures, there are RLRs, NLRs, and TLRs that help us. These are the PRRs. Okay, so that help us to basically recognize the pathogen. Okay, so we have in the RLRs we have RIG1, MDA5, and LGP2 that acts through the adapters, that is MAVS. Okay, so this is an adapter that functions in relation to all these receptors. We have in NLRs we have N node 1, node 2, NLRC4, NLRP1, NLRP2 to 40. Okay, and the adapters for different are ASC. Then we have NLRP3 inflammasomes. Okay, the procaspase one. So this forms the NLRP3 inflammasome that is a major contributor for inflammation. Then we have the TLRs. So TLRs basically help in work by MyD88 and TRIF pathways. Okay, and there are some other molecules also, that is AM2 and P202 that function with AM2 ASC, that is AM2 inflammasome that is formed. Okay, so these yellow ones are the intrinsic innate cell intrinsic innate immune sensors, so they are present inside the cell, and the green ones are the cell extrinsic innate immune sensors, so they are present on the surface or outside that. Okay, so these are the RIG1 and MDA5 virus recognition pathways. Those are RLRs basically. Okay, so what will happen? So RIG1 and MDA5, they recognize, so RIG1 recognize the single stranded RNA that have the 5 prime triphosphate, and MDA5 has a long strand of double stranded RNA. So now these are the viruses that are recognized by this one is Sendai, vascular stomatitis, influenza, hepatitis C, Japanese encephalitis, small RNA encoded by the DNA virus, Epstein-Barr virus. Okay, so these are the major viruses that are recognized by the RIG1. Now, for the MDF5, dengue virus and West Nile virus are recognized by both RIG1 and MDF5. Okay, Now, from this RIG1 and MDA5, it leads to MAVS. Now, MAVS it activates various inflammatory pathways like TRAF6, TRAF3, and FADD RIP1 pathway. Okay, so all these pathways leads to NF kappa B activation, and NF kappa B is a transcription factor that leads to production of the cytokines that are pro-inflammatory. For example, IFN beta and all the other cytokines that are pro-inflammatory, that are uh, basically transcribed by the NF kappa B pathway. Okay, so this is how the RIG1 and MDF5 leads to the production of NF kappa B, and this NF kappa B leads to the activation of different cytokines so that they can go and activate the inflammatory pathway. Okay, then the picorna virus and can be activated by the cytosolic synthetic double-stranded RNA, and it has a polycynic polycystidylic acid. Okay, so that is a synthetic RNA that can be activated, that can activate this pathway. Okay, so it can be used as a vaccine against picorna virus. Then the MDA5 also recognizes large double-stranded RNA structures that lack 2' O methylation generated in the cytosol during virus infection. So this is also recognized by MDA5. So these are all part of the innate immune system, and that through RLR's pathway. So this is the first pathway how a virus is recognized by the innate immune system. Now moving on, we have the TLR pathway. Okay, so the TLRs that are functional for virus recognition in mice are 1 to 7, 9, and 11 to 30. Okay, then in the humans we have TLR 1 to 10, out of which only 10 is functional. For the cell surface ones, we have TLR 1, 2, 4, 5, 6, 11, and 12 that are functional, and present on the endolysosome is 3, 7, 8, and 9 that recognizes the viruses. So what happens is the these TLRs that are present on the endolysosome, TLR 3, 7, and 9, they recognize the virus that enters through the endosome. So they are present on the endolysosome, and they recognize the viral patterns that are in that actually enters through the viral cells, and then they go through the reverse pathway for the production of more TLRs. That means uh, so so what happens is there are TLR transcription that so this basically figure shows that the TLR processing and activation on the endolysosome is done by an important gene, that is UNC93B, that is encoded by the gene UNC93B1 that are found in. And if there is any mutations in this gene, then the HSV encephalitis, it has been found in two patients that have severe encephalitis, HSV encephalitis, that have a mutation in this gene, because this gene helps in activation and the production of the TLRs 3, 7, and 9 on the endolysosome. Okay, so these are important in recognizing the viruses in the endolysosome. Okay, now whenever they recognize any of the viral signatures in the endolysosome, so this is TLR 3, 7, or 8, and this is 9. Okay, so they activate like the RLRs, they activate the TRAP3, TRAP6, MyD88 pathways. Okay, so these two are common with the last one, and these TLRs basically function with the MyD88 MyD88 adapter. Okay, so this further activates the NF kappa B path. the same pathway. Just the receptors are different for the different receptors for the innate immune system. Okay, now they increase the production of the NF kappa B that leads to the signaling of the inflammatory cytokines. Same is the case with IRF7. So IRF7 leads to the production of IFNs, that is interferons. Okay, so TLR3 basically uh, recognizes the DNA viruses and those DNA viruses that can produce a double-stranded RNA. Okay, so in if there is any dominant negative mutation in the TLR3 gene, then there is neonatal herpes infection. So that means in the if there is any mutation in the TLR3 gene, there can be neonatal herpes infections because TLR3 is specifically involved in recognition and inhibition of the herpes virus. Okay, then TLR7 is basically present in the peripheral dendritic cells, except for CD8 alpha positive dendritic cells in the B cells, and it senses influenza, SEV, and the VSV. Okay, so these are the viruses that are being recognized by the TLR7. And mouse TLR8 is expressed by myeloid disease and the monocytes. So TLR7 and 8 both recognize a single-stranded RNA as we studied before. The uridine and the ribose defining the signature of the RNA are both necessary and sufficient for TLR stimulation. So whenever there is an any RNA, if there is uridine, that is uracil present, and the ribose along with that, so in that case, it gets activated and then it leads to the stimulation of the TLR7 pathway, that is through TRIF pathway, and leads to the activation of the NF kappa B pathway. Okay, now the viral fusion and uncoating and the endosomal acidification are required for the TLR dependent recognition. So because it is present on the endosome, so what will happen? The acidic condition, the viral fusion or uncoating, and the endosomal acidification both are necessary for the TLR7 dependent recognition. In the literature, it suggests that TLR8 deficient mice develop autoimmunity due to hyperstimulation of TLR7. Okay, so this can be the case where TLR8 develops autoimmunity because of TLR7. Okay, so this is how, and now we have the function of the TLR9. Okay, so TLR9 is expressed in the peripheral dendritic cells and the B cells. In the mice, it is expressed in peripheral D cell, B cell, and the macrophages and conventional DCs. Several viral DNA and bacterial DNA sequences containing hypermethylated CpG motifs, they recognize the HSV1 and HSV2, that is the herpes simplex virus 1 and HSV2. So recent studies showed that TLR9 recognized the sugar-based backbone, 2' deoxyribose of phosphodiester DNA, and irrespective of the CpG content. Okay, so they are majorly recognizing of the double-stranded DNA. So they recognize the sugar-based backbone and the 2' deoxyribose sugar. Okay, so this is how TLR9 functions. So this was the part where the innate immune system got active and recognized the viruses. Now we'll see how a microRNA basically helps in recognition and inhibition of the viruses. Okay. So microRNAs are basically non-coding RNAs that are present in the, so these are non-coding RNAs that basically helps in the regulation of the gene expression of various genes inside the body. Okay, so this is the pathway how a microRNA gets is expressed or produced inside the body. So the microRNA gene is this one, and RNA polymerase 2 or 3 leads to its transcription. They produce a primary microRNA. Okay, so the primary microRNA is cleaved into pre-microRNA that from the DROSHA or DGCR2, DGCR8. Okay, this after this gets exported into the cytoplasm, and here the exportin 5 and the RAN GTPase helps in its export. Now the pre-microRNA gets a cleavage from the dicer, and it is formed into a microRNA duplex. Okay. Now this duplex, out of this, the passenger strand is basically cleaved, and it leads to its degradation. Now after this, the major target strand uh, is complex with risk formation, that is a complex, okay, with the argonaut 2 protein, and the mature microRNA gets activated, and it leads to the mRNA target cleavage, translational repression, and the mRNA deadenylation. So these are the some of the targets that how a microRNA functions. Okay, now we'll see some of the examples how a RNA leads how a virus leads to the production of the microRNA that helps in its infection, or how a microRNA actually resists its infection. Okay, so here is an example of an RNA virus that leads to the activation of the rig1 protein, that is from the RLRs, and it leads to the production of the type 1 interferon and the inflammatory cytokines, so that induce an antiviral response. Now there are host microRNA 485. Okay, so these microRNA directly bind to the rig1 mRNA and leads to its suppression. So it degrades the rig1 mRNA. Okay, so what will happen? Now the rig1 receptor is actually down regulated. So because of this down regulation, it leads to enhancement of the viral replication, and it leads to the increase in the load of the virus. Okay, so as we can see here, if the mir 485 is actually reduced, so in that case what happens, the level of the influenza virus actually reduced after 24 hours in this experiment. Okay, so this is how a microRNA help the virus to infect the or actually induce an antiviral response. Okay, now, moving on to another example, this is from a highly pathogenic avian influenza virus, that is H5N1. Okay, so what happens is, in H5N1, the viral genome can constitutively express in the SUDEC2 mRNA, that is a negative regulator of the JAK-STAT pathway that reduces the viral load. It inhibits MIT 324-5P. So what will happen? This MIR 324-5P leads to the enhanced viral replication. That means it helps in immune evasion from the influenza virus. Okay, so it inhibits both JAK-STAT and the PB1 genome in the PB1 gene in the viral genome. Okay, so it inhibits both of them, and this leads to reduction in the sorry. So whenever there is reduction in the MIR324-5B, that leads to enhanced viral replication. Okay, so because this MIR is suppressing the JAK-STAT pathway and the PB1 genome, PB1 gene in the viral genome. Okay, so if it suppresses both of them, that leads to reduced viral load in the body. Okay, so because of this, there is no much infection of the influenza virus. But if this MIR324-5P is reduced after the viral infection, that means it will lead to the more infection of the viral particles. Okay, because it will not be able to inhibit the host cellular machinery. Okay, so this is how the viruses actually got help from the microRNAs. Okay, now we'll see certain innate immune system evasion strategies, how a virus actually evades the immune system. Okay, so we have the virus host interaction, that means the evasion of the innate immune system. So all the viruses that encode factors to counteract the induction, signaling, or the anti-pathogen effector responses. Okay, so that they can hide from the immune system and they can activate their processes. So they employ distinct molecules to achieve blockade of the antiviral effects of the type 1 interferons. These are categorized into four major categories. Okay, global inhibition of the cellular gene expression. So the cellular gene expression is reduced after a viral infection. Evasion of the innate recognition, that means they can hide themselves from the RLRs, TLRs, or NLRs. Okay. Inhibition of molecules involved in the IFN induction and signaling. Inactivation of the IFN induced effector molecules. Okay, so these are the four major techniques how a virus evades its immune system. Okay, so the inhibition of the global gene expression, evasion of the innate recognition, inhibition of the molecules that involved in IFN's induction, and inactivation of the effector molecules of the IFNs. So one of these is cytoplasmic helicases. Okay, so what happens is there are cytoplasmic helicases that are present inside the cytoplasm that help in deactivating the particular RNAs that are produced by the host cell to circumvent the IFN protein production. Okay, so this is one of the techniques takes place in the system. Then we have the toll-like receptor technique. So in that case, what happens? The TLRs basically helps in the, as we discussed previously also, it leads to the MyD88 or TRIF based pathways to increase the inflammatory response. Okay, and then we have the signaling about these for the for the production of the IFN proteins and the antiviral effectors that leads to the suppression of those viral particles and increase in the cytokines so that they can reduce the viral infection. Okay, I have not gone into much detail because that is out of the scope of this particular presentation. But these are the major pathways that are recognized by the viruses to circumvent the innate immune system. Okay, now how does the adaptive immune system helps in the virus recognition and its lowering? Okay, so this is the virus. There are two pathways through which the neutralization takes place. First is the neutralizing antibody, and the second is the complement mediated lysis and phagocytosis. Okay, so its infection leads to the production of the neutralizing antibodies against the infected cell. The then there are infected cell lysis or clearance using FC mediated, basically ADCC. Okay, then we have the cell signaling to inhibit the viral replication. Then we have steric obstruction to inhibit the viral release, and then the cell to cell transmission. Okay, so these are the pathways how a system basically inhibits the virus release and its infection to the whole body. Then the what happens is the effectors basically target the CD8 plus and CD4 plus or CD4 positive T cells to activate, and the effector mechanisms are continue. Okay, for example, these are the cytotoxic T cells. Now, whenever there is a direct infection by the on the cell, so what happens? The macrophages or the phagocytes, the antigen presenting cells, they take up those virus infected cells, they neutralize them, and they present the antigens in the form of MHC class 1 to the CD8 cells. Okay, so what they effect is direct cytolysis, that means cell to cell contact, granule exocytosis through perforin and granzyme, facile, that is a facile facef interaction for the cytokine release, and IFN gamma and TNF alpha or the cytokimokines to release. Okay, so this is for the inflammatory response. So this is the how the CD8 positive cells affects for the virus infected cells. Okay, so there is a regulation also here. IL-10 damps the CD8 T cell responses. That means IL-10 basically helps in regulating the CD8 positive T cell response. Now we have the CD4 positive T cell, that is the T helper 1 cells. It functions with the class 2 molecules. So what will happen? Now the class 2 molecules will directly present those cells through the uh, to the CD4 positive T cells, and it will release the IFN gamma, TNF alpha chemokines, the cytokine synthesis and its release. Okay, same is with the Th2, that is the other population of the helper T cells. These release cytokines like IL-4, IL-5 and IL-30. Then Th17, those are termed as the regulatory cells. So they also release cytokines that are 17AF, IL-6 and IL-21. So these are the different cytokines that are produced by the different cells so that they can circumvent the viral infected cells. Okay, then we'll see the memory BNT cells generated after infection or vaccination are generally long lived. So now, because the reinfection of these particular viruses can lead to immunity. So now the factors that control the formation or duration of the memory immune responses are not well understood, but what will happen? The B cell activating factors play an important role in maintaining the viability and the basal homeostatic proliferation of the memory cells. Okay, so now what will happen? There is a plasma cells. Okay, so that produce the antibody, and these plasma cells are basically They have two types of plasma cells. First is the memory plasma cells also. And what will happen because of these memory plasma cells, some of the viruses have a long activating or long sustaining plasma cells that leads to the production of antibodies for a long term of long term. Okay, so after the infection also the body recognizes those viruses, and because of the antibodies that are produced by those long term plasma cells. Okay, now, the two mechanisms that exist are repeated exposure to the antigen so that the memory B cell levels get maintained, or the pre existing serum or tissue high affinity, the class switched antibody to the virus, and the long lived plasma cell in the bone marrow. Okay, so these are the two basic mechanisms of how a virus infected cell or a immunity functions. Okay, so the this immunity, the memory cells actually how they functions. Okay, so these are the long-lived plasma cells that I was talking about in the previous slide. They secrete high-affinity antibody and survive without cell division for many years in humans, and they contribute to the maintenance of antibody level over decade. Okay, so these depend on the antigen independent or the bystander activation because they'll be producing antibodies for a long period of time. Okay, then we have the short-lived plasma cells. They survive only for a few days. They produce antibody in extrafollicular foci, and they will be crucial in very early response to the pathogen. For example, here we have in Sweden, the inactivated poliovirus showed sustainable anti-poliovirus antibody titers in all age groups. Okay, but the maintenance of the antigen antibody titers over decades in the absence of further vaccination or exposure to the live virus. So that means they are sustained for a long period of time. Okay, so long period can be decades, 2 decades, 3 decades, etc. But in case of tetanus or diphtheria, what happens? The antibody titers declines. So that means they do not have a long-lived plasma cells. Okay, so that means they'll not be able to produce antibodies upon reinfection. That's why for the tetanus vaccine that we take, that we have to take every 6 months or every 1 year if there is any wound or something, because we will not be able to have those antibodies present in our body so that they can be used for the reactivation. Then we have the smallpox vaccination using vaccinia virus. So what happens is the peak responses drops at after the vaccination over a 10-year time point, but stable levels were seen between 10 to 16 years. Okay, so and the serum antibody responses could be identified at 60 to 75 years after vaccination also. Okay, so what happens is in case of the vaccinia virus vaccination for the smallpox, what happens, there is increase in the antibody titer once, then after 10 to 5 years the antibody titer becomes stable. Okay, so this is because the serum actually have these antibodies, and some of the plasma cells are maintained inside the body. Okay, although the mechanisms that keep these plasma cells circulating for a long time is still yet not known in detail, but there are the composition of the viral particles, the induction of the immune response also plays a major role here. Okay, so now moving on to the this was for the B cell. Now moving on to the T cell memory. Okay, so what happens? The activated naive T cells, give rise to both effector T cells and the memory T cells. Okay, so T effector or the TE and T memory or the TM. The difference or the relationship between two naive T cell products is unclear. That means there is differentiation in the expression of CD45 only yet. But we have to explore more on the effector and the memory T cell, how they actually function and what are the structures that are present on them. Okay, the T effector cells have high frequency and their lower activation threshold to trigger cell proliferation and differentiation. Memory T cells can also rapidly express effector activity, example pro-inflammatory or antiviral cytokines such as IFN gamma or TNF alpha, within hours after TCR engagement on these T cells without DNA synthesis. That means additional cell proliferation. Okay, so this is basically lead to the whenever there is TCR engagement on these T cells, so these leads to effector activity of the particular virus infected cells. The naive T cell activation and proliferation in memory T cell population formation and maintenance are controlled by three cytokines 2, 7, and 15. Okay, so three cytokines are involved: IL-2, IL-7, and IL-15. So, for the T cell activation, naive T cell activation, proliferation, and maintenance of the memory T cells. IL-15 dependent and long-term memory T cell viability. Suppression of the apoptosis is supported by IL-7, which upregulates or sustains the expression of anti-apoptotic BCL2 gene family in the developing memory T cells. Okay, so IL-15 basically helps in long-term memory T cell viability and is supported by IL-7, which regulates or sustains the BCL2 apoptotic gene family members. Then they are developing memory T cells. So BCL2, that is the major pathway for the anti-apoptotic signals. So these leads to the T cell viability. Okay. Memory CD8 plus T cells differentiation. How they differentiate? So the basically, so what happens is, this is a naive T cell, CD8 positive. Okay, so whenever a pathogen encounters to this leads to the formation of effector T cells and the memory T cells. These memory T cells basically are uh, sustained in the body for a long time. They are functional and they sustain for a short period of time. So they are short lived. Whenever there is re-encounter of the pathogen, this TM again converts to TE and TM. Okay, now this T will function at that time and TM will have that memory of the second infection also. Okay, so these memory T cells self renew into the memory T cells again, so that they have extended lifespan that depends upon IL-7 and IL-15, depending upon the homeostatic proliferation, how slow cell division and minimal cell number changes. Okay, so the same thing is explained again and again. Now we'll be moving on to how the virus basically helps in evading the innate immune or adaptive immune system. Okay, so they escape by a mutation. So, for example, the recent example we'll take coronavirus. Okay, the initial infections were not that aggressive, but after the second wave, the infection became aggressive because of the mutations. Okay, so those mutations are not recognized by the adaptive immune system of our body because of those mutations in the structural protein genes. Okay, then escape by latency. So latency means the virus will sit in the body for a long period of time, and then it will reappear whenever it will get the favorable conditions. Then it can escape by destruction of the immune cells. So yes, obviously, like in HIV, it directly targets the T helper cells, okay, or the macrophages. So it will directly destruct the immune cells, so that it can infect the other body cells. Then they can control the antigen processing and presentation machinery, inhibit the T cell mediated targeted lysis, inhibit the inflammatory responses or the cytokine action, or inhibit the humoral immunity by virally encoded Fc receptors. Okay, the complement receptor or the control proteins. So this is how the several ways how a virus can escape the particular uh, T cell responses. Okay, the then we have the inhibition of the class 1 or class 2 MHC complexes. That means the antigen processing and presentation. Okay, so for example, we have the HIV TAT protein that have been reported to suppress, inhibit MHC gene promoter activity. So it decreases up to 12 times. Then we have human cytomegalovirus that impairs MHC class 2 expression, escape by subverting so MHC class 2 presentation is impaired by the human cytomegalovirus. Okay, so these are some of the steps that are used by the viruses to circumvent any of the MHC1 related pathways. So, if we have the MHC1 class 1 synthesis inhibition that is done by the lentivirus VPU protein, we have the TAP, that is transporter associated with the antigen processing, its expression and function. The expression is inhibited by EBV and HCMV, that is cytomegalovirus. These are the proteins that help in that. For the function, we have the for the CMV, it's US6, and HSV we have ICP47. For inhibition of the MHC1 class 1 transport, we retain MHC class 1 in the ER done by the adenovirus or the CMV. Pre-Golgi compartment, that is done by the CMV M152. Dissociate MHC class 1 to the cytoplasm by US11 and US2 proteins of the HCMV. Disclose MHC1 class 1 to lysosome. This is done by M6 or GP48 from the cytomegalovirus. Then GP34, it bind to cell surface of the MHC class 1 molecules. That means it uh, do not allow the MHC1 class 1 to function. Then increase the endocytosis of the MHC class 1 molecules, that is NEF, K3 and K4 proteins of the HHV and the HIV. Okay, so this is how different proteins from different viruses affect the class 1 synthesis and its activation and the transport of the MHC molecule. So now let's move on to the viral modulation of the cytokine system. So what happens is there can be two ways that the virus can modulate either the cytokine function or the cytokine production. Okay, so there are three ways basically. So either they can interrupt with the cytokine production, that means they can interfere with synthesis or their generation of the functional cytokines. Okay, so that means either the synthesis of the cytokines or their functional components, right? So this was the first case. Then in the second case, they can interfere with the cytokine action. This is a main role of the viral modulation. Okay, so they can encode homologs of the cytokine or chemokine receptors, like the vaccinia virus basically encodes for a type 1 interferon homolog, and interferon gamma homolog is also produced by another strain of the vaccinia virus. Interleukin 6 homolog is produced by KSHV. Then in IL-8 homolog is produced by human cytomegalovirus. IL-10 homolog is produced by EBV, that is Epstein-Barr virus, and the cytomegalovirus. Then there are certain soluble cytokine receptors to neutralize cytokines. These are produced like IFN gamma receptors. These are produced by myxoma virus. Then IL-1 beta receptor as produced by vaccinia virus. There is another strain, and the TNFR, that means tumor necrosis factor receptor, that is orthopox virus. So these are the different uh, viral modulation techniques, that means that can interfere with the cytokine action. And the last one is they can affect with the cytokine effector function, that is cytokine signaling pathways. Okay, and like the cowpox virus, CPV, they basically there is a cytokine response modifier, a CRMA, that inhibits the production of caspase 1. That the caspase 1 is basically a modulator of the inflammatory pathway, which prevents the proteolytic cleavage of pro interleukin 1b to mature 1 interleukin 1 beta. Okay, then the myxoma virus, it produces VNT, V viral TNFR, and the viral IFN gamma receptor, or the vaccinia virus also produces IFN alpha or beta receptor. Okay, so these are some common examples of how a virus can modulate the immune system. Then the viral evasion of the humoral immunity. So now, till now, we have discussed about the till now we have discussed about the different ways how it can modulate the cytokine system. Now we'll move on to the B cell immunity or the humoral immunity. Okay, so the viral strategies that are being used to evade the neutralizing antibodies, responses to the viral surface protein, so they can actually mutate the surface proteins or by antigenic drift or shift and recombination, so that they can produce variable viruses. For example, we have influenza virus, like we have H1N1, then we have H5N1. So like this is a common basic example of the influenza virus that gets mutated. So the hemagglutinin in the influenza virus, that stands for H. So it gets mutated and it leads to variable viruses. Okay, so that actually escape from the neutralizing antibodies. Then the masking of the epitopes. Okay, so for example, this is a epitope that is produced by the original virus against which the antibody has been produced. Now it changes to this. So now the antibody cannot identify this particular epitope. So this is how they mask their epitopes. Then glycan shielding. So glycan shielding is basically the shielding by the carbohydrates. Okay. Then surface protein decoy. That means uh, they can mop up the neutralizing antibodies you under certain conditions as seen in HBV. Okay, then original antigenic sin as seen by the HIV and the influenza virus. The irregular surface protein spacing that tends to reduce the immunogenicity. So they can change the epitopes depending upon the spacing on the surface. Okay, then multiple surface proteins and the virus forms. Uh, that means there are different kind of proteins on the different on the surface as well as different virus forms. So they can encode multiple proteins for the entry. Okay, so this is how they can evade the humoral immune response. Now, how this can actually lead to the sorry, how this can actually lead to the autoimmune diseases. So in that case, what happens, the viral infections, particularly the chronic ones, they enhance the autoimmune disease susceptible to in susceptible individuals. because of their strong inflammatory response in various organs. Okay, so major pathways that likely to cause the autoimmunity are the direct infection of the target organs or cells that can cause the release of sequestered autoantigen and enhance the antigenic presentation. Local inflammation that might alter the repertoire of self-epitopes presented by the APCs by altering antigen degradation properties of the proteome, and presentation of the pathogenic epitopes with structural or sequential similarity to self-epitopes that might activate the autoreactive lymphocytes without mimicry. That is molecular mimicry of the particular epitopes that are self-antigens, recognized as self-antigens, but these are presented by the viral subunits. Okay, so these were the some of the strategies how a virus can actually evade the immune system by acting in either of the forms. Okay, now we'll move on to the questions. So the first question is, which characteristic distinguishes viral nucleic acid from the host nucleic acid, facilitating pathogen recognition receptors recognition? So what can be the correct answer? So the options are presence of long double-stranded RNA structures in the cytosol, high levels of 3' triphosphate on the RNA molecules, highly methylated CpG motifs, exclusively nuclear localization of viral nucleic acids, and frequent presence of polyadenylated tails on the viral RNA. So the correct answer is option 1. So they recognize the long double-stranded RNA structures that are being sorry, that are introduced into the cytosol of the that are introduced into the cytosol of the particular host cell from the RNA molecule. Okay, so this has been recognized by the PRR recognition. Okay, now moving on to the next question. Which mechanisms are known to be triggered by type 1 and type 3 interferons as part of the antiviral response? So, what can be the correct answer? So, let's see the option. Induction of apoptosis in the induction cell. So the options are induction of apoptosis in infected cells. Yes, this is the correct answer. So they actually they actually lead to apoptosis of the infected cell. Then direct neutralization of the viral particles. No. Activation of the interferon stimulated genes. Yes, this is correct. They tend to produce the interferon stimulated gene so that they can produce a inflammatory response. Okay. Enhancement of host cell protein synthesis? No. Activation of the antigenic specific cytotoxic T cells? No. Then downregulation of the viral regulation pathways? Yes, this is correct. Recruitment of the professional phagocytes? Yes, this is also correct. Then stimulation of the B cell proliferation? No, this is false. So the correct answer is 1, 3, 6, and 7. Okay. So let's move on to the next question. Which of the following mechanisms involve evading neutralizing antibodies by altering viral surface proteins by variable viruses like influenza virus or the sarcoma viruses? So, what can be the correct answer? So the options are molecular mimicry, antigenic drift and shift, complement inhibition, glycan shielding, and decoy receptor formation. So here, because the example is influenza virus or HCV, so in that case, what happens? There are different forms of the virus that we called as strains. So what they strains do? They change their antigen present on the surface. Okay, so they change this antigen that is present on the surface because of the mutations. So the mutations or recombination that we call. So this is called as antigenic shift or drift. Okay, so the correct answer is option number two. That is antigenic drift and shift. Okay, so let's move on to the next question. Which virus is specifically recognized by RIG1 despite having a DNA virus? So what can be the correct answer? So here the correct answer is Epstein-Barr virus. That is option number 5. Okay, so it is only recognized by RIG1 despite being a DNA virus. The other viruses are RNA viruses that are recognized by the RIG. Okay, so let's move on to the next question. What role do cytotoxic T cells, CD8 positive T cells, play in the adaptive immune response? So what can be the correct answer? So the options are they exclusively produce antibodies against the pathogens. No, this is the work for B cells. Okay, they rely solely on antibody mediated neutralization. No, this is also false. They induce apoptosis in infected or abnormal cells through perforin and granzymes. Yes, this is correct. Okay, they activate B cells without the need of the cytokines. No, they suppress the immune response to prevent autoimmunity. So this is also not correct. So the correct answer is option number three. They induce apoptosis in the infected or abnormal cells through perforins and granzymes. Okay, so let's move on to the next question. The next question is what is the key strategy viruses uses to evade the immune system? So what can be the correct answer? So the options are they enhance immune signaling to avoid detection. No, this is false. They suppress that or destroy the immune cell to escape immunity. No. They completely block antigen presentation in all cells. No. They increase antibody production to overwhelm the immune system. No. They prevent cytokine release without affecting other immune functions. Oh, so the correct answer is option 2. They suppress or destroy immune cells to escape the immunity. Okay, so let's move on to the next question. What key concept does the myxomatosis experiment demonstrate in host pathogen interaction? So, what can be the correct answer? So, the options are host always evolves to completely eliminate the pathogens. No, this is false. The pathogens often become less virulent over time to maintain survival within the host population. Yes, this is correct. The pathogen increase in virulence over time? No. Host become genetically identical to resist infection? No. Viruses can no longer mutate once inside the host? No. This is also false. So the correct answer is option number 2. Okay, so let's move on to the next question. What role do small interfering RNAs play in antiviral defense? So what can be the correct answer? So the correct answer is option 1. They recruit the risk complex to clean the viral RNA, providing sequence specific defense from the immune system and the human and the host body. Okay, so let's move on to the next question. Which of the following are viral strategies to evade the host's innate immune response? So the options are global inhibition of the host gene expression. Okay, so yes, this is correct. Okay, so they can actually evade the global gene expression strategy so that they can produce their own viruses. Okay, suppression of type 1 interferon signaling. Yes, this is also correct because the type 1 interferon is the major cytokine that leads to the viral response, inhibition of the viral response. Inhibition of the pattern recognition receptors. Yes, because they tend to identify the viruses. Activation of the transcription factor to enhance immune response? No. Increased activation of the adaptive immunity? No. Targeting of immune response stages from detection to transcription? Yes, this is correct. Then deactivation of the co-stimulatory signals? No. Suppression of host transcription machinery? Yes, this is also correct. So the correct answers are 1, 2, 3, 6, and 8. This is how the virals actually evade the host immune responses. Okay, then let's move on to the next question. Which of the following statements are correct? So the options are dengue virus and West Nile virus are recognized by both RIG1 and MDA5. Yes, this is correct. Okay, both are so because both are the RNA viruses. Okay, RIG1 recognizes large double-stranded RNA structures lacking 2' O methylation generated in the cytosol. Yes, this is also correct. So this is false. P coronavirus can be activated by cytosolic synthetic dsRNA, poly in silico in silic polycyclic acid, that is poly-IC. Yes, this is correct. The host DNA but not viral DNA can be present in the cytosol. No, this is false. So only the viral DNA can be found in the cytosol but not the host DNA. PRRs are present in the influenza virus genome. No, this is false. DNF alpha is an inflammatory cytokine. Yes, this is correct. DNA viruses can produce double stranded RNA. Yes, this is also correct. TLR7 can sense influenza virus RNA. Yes, and the TLR8 deficient mice can develop autoimmunity due to hyperstimulation of the TLR7 IgA. Yes, this is also correct. So the correct answers are 1, 3, 6, 7, 8, and okay, so let's move on to the next question. Which of the following statements are correct? So the human microRNAs can bind to viral RNAs. No, this is false. So this is not correct. Okay, so yes, this is correct. The microRNAs predominantly target 5' UTRs of the mRNA. Yes, so this is false basically. Okay, primary microRNA is produced in the cytoplasm. No. Okay, IL-10 damps the CD8 T cell responses. Yes, this is correct. Memory T cells always take long time to express its effector activities. Yes, this is correct. Okay, because sorry, this is false. Uh, Memory T cells actually take less time as compared to the effector T cells because that is a that is due to the secondary immune response. Okay, then naive T cell activation is mediated by IL-2. No, this is correct. HIV tag proteins induce MHC gene promoter activity. No, this is false. HCMV can impair MHC gene class 2 expression. Yes, this is correct. Repeated exposure of antigen in one way in which memory B cells level can be maintained. Yes, this is also correct. So the incorrect options are 2, 3, 5, and 7. Okay, so let's move to the next part, that is the introduction to the influenza virus. So, the influenza virus is one of the key factors that were involved in the Spanish flu pandemic. Okay, so that actually emerged during the 1918, the most severe pandemic that was called as the Spanish flu, and it spread worldwide during the 1918 and the 1990s. And around 500 million people, or the one third of the world's population, was affected. Okay, the number of deaths included 50 to 100 million, that is 5 to 10 crore. In India itself, the deaths were 10 to 20 million. Okay, mortality was high in people that are younger than the 5-year-old, or the 20 to 40-year-old people, or the older age ones, because they have the immune system compromised. Okay, or because of the age. Now, as you can see here, this is a bird, that is the aquatic bird, that is the natural reservoir of the particular virus. Now from this, if it affects any of the other species or any other animal of the animal kingdom, so in that case it will called as a zoonotic virus, and it can actually spread to avian birds that can fly, or any of the sea organism other than the aquatic birds, then any of the birds that stay on the ground. For example, here we can see a hen, a chick. Then any of the animals that stay on the ground, for example, here the horse, pig, and cat or dog, and ultimately to the humans. And if any of the viruses are spreads through the uh, spreads through the animal virus, then that is called as a zoonotic virus, and that spreads very highly in case of the human population, because we do not have any immunity against these viruses. Okay, and the immune system also takes more time to develop immunity against these viruses. Okay, so let's see about the structure of this particular virus. So it has a negative sense single stranded RNA, and that is spherical or ovoid in size, ranging from 90 to 100 nanometer, and have a segmented genome. So segmented means it has 8 different segments of the single stranded RNA, that is between 13.6 kb. So the whole genome, total genome is comprised of 13.6 kb. As you can see here, this is the particular structure of the virus. So we have the capsid that have the neuroamidase and the hemagglutinin. So that gives its virus name, that is H1N1, for example. Okay, so H stands for hemagglutinin and N stands for neuroamidase. So that helps in viral infection basically. Then inside it we have the matrix proteins. Okay, so this is a matrix protein 1 and this is the matrix protein 2. Then we have certain a layer of mm, then we have the nucleoproteins or the nuclear export protein and the viral ribonucleoprotein that helps in the the production of the viral RNA, viral DNA from the viral protein from these particular RNAs. Okay, so let's see the genome segments that are present in the particular virus. So first we have the PB2, that is around 2 kb. It produces a PB2 protein. So PB2 protein is a component of the RNA polymerase that recognizes the cap. Okay. Then we have the PB1 protein. This also is around 2.3 kb, and it produces two proteins, PB1 and PB1F2. Sorry, three proteins, PB1 N40 also. So PB1 is a component of the RNA polymerase that helps in elongation, and PB1F2 has a pro-apoptotic activity and is an IFN gamma antagonist. So it can stop the activity of IFN gamma. Okay, and the PB1N40 is of unknown function. So we don't know the function of the PB1N40. Moving on to the third protein, that is PA. This is around 2 to 2.2 kb. It produces different proteins linking to the PA, and these are the components of the RNA polymerase that have the endonuclease and the protease activity. Then comes the fourth component, HA 1.7 kb. This produces hemagglutinin, that is a surface glycoprotein and a receptor binding that have fusion activity also. The major antigen and assembly and binding also is taken care by the HA. Then we have NP, that is 1.5 kb. So NP is a nucleoprotein. So it has RNA binding, RNA synthesis, RNP import, nuclear import properties. Okay. Then we have the Na, that is neuroamidase. So neuroamidase is 1.4 kb. The gene is 1.4 kb, and it is a sulfase glycoprotein that have a neuroamidase activity. So it can cleave the protein that help in binding as well as inclusion in the endosome. Okay, then we have M protein, M gene. M is around 1 kb. It encodes for M1, M2, and M42. So M1 is a matrix protein that interacts with the RNPs and the glycoproteins, and it has RNP nuclear export and the assembly and binding process. Then M2 is also a membrane protein. It is an ion channel that helps in maintaining the pH of the particular capsid. Okay, so it also helps in assembly and binding. Now the last one we have the NS. So NS is around 800 base pairs, and it encodes for NS1 and NEP or NS2. So NS1 is a multifunctional protein that is also an IFN gamma antagonist, and NS2 is the RNP nuclear export regulation of the RNA synthesis. Okay, so these are the eight components or the eight segments of the viral genome that encodes for different proteins, include involved in RNA polymerase, the maintaining of the capsid, fusion of the endosome and the fusion of the endosome and the virus, as well as its nuclear import. Okay, so now Let's move on to naming of the viruses. So the name is given by the antigenic types, the host origin, that is swine, equine, or chicken from which it's originated. For human origin, no host of the origin designation is given. For example, in the duck, avian influenza A H1N1. So A duck slash Alberta, the place. Then this is the strain number, and this is the year of collection. Okay, so this is how it's written for in humans. If it is organized, it is originated, then we give the number as this. Okay, then we have the geographical origin where it was first identified. We have Denver, Taiwan, etc. Then we have the strain number as 7 and 15, and then we have the year of collection. So this is how a influenza virus is named. For influenza A viruses, hemoagglutinin and neuraminidase antigen description are provided in parentheses. For example, influenza A H1N1. So this is the uh, identification of those influenza virus. Then we have another influenza virus H5N1. So this is another influenza virus with the same strain. In 2009, the pandemic virus was distinct name H1N1 PDM09 to distinguish it from the seasonal influenza virus AH1N1 viruses that circulated prior to the pandemic. Okay, so when humans are infected with the influenza virus that normally circulate in swine, these viruses are called variant viruses and are designated with the letter V. So, example, if this is V is present, that means that will infect swine as well as humans. If the virus is isolated from a non-living material, for example, from soil or from water, then the nature of the material is specified. For example, here the lake water is mentioned. That means it was isolated from the lake water. Okay, so to brief, A is the virus type, the place where it is isolated, the strain number, and where it was isolated, and this is the virus subtype. Because it was isolated from humans, it is not specified here. But if it would be isolated from a different species, then it would be specified here. Okay. Then we'll move on to the last question for today. Which of the following proteins in the influenza virus genome is primarily involved in the inhibition of type 1 interferon? So, what can be the correct answer? So, the correct answer is option number 3, that is NS1. Okay, so this is the uh, protein that helps in inhibition of the type 1 interferon, the primary protein that helps in its inhibition. Okay, so that was the last question for today. Thank you guys. If you guys have any doubt, you can ask. If not, then we'll close the session for today. Thank you guys for joining in. We'll see you next week.